Hello, everyone. Welcome to Chemistry with Professor John Nguyen. But today we finishing up the biochemistry part with nucleic acids. These are very important molecules. They are molecular blueprints um, that control the synthesis of proteins. Okay, and they ensure that the protein have the correct amino acid sequence. Okay, these amino acids. Well, nucleic acids contain chemical code that specify the correct amino acid sequences for protein. And there are two different types of nucleic acid. That's DNA, which exists in the nucleus of the cell, the center of the cell, and um, RNA stand for ribonucleic acid, which is found in the cytoplasm throughout the entire interior of the cell. Okay, just like protein, nucleic acids are polymers of single units known as nucleotide. All right, we'll look at that in a minute. All right, now each nucleotide is a unit of it's a monomers of DNA or RNA. Okay, each nucleotide, the first component is the sugars. In DNA, we have deoxyribose sugars. These are the monosaccharides. Now in RNA, we have ribose sugars. Notice the difference is the OH. In deoxyribose for DNA, there's no oxygens. That's what we call deoxy, meaning missing the oxygens. All right. Now, the other two components are the base and the phosphate. And so if you got DNA, you have the sugar, the base, and the phosphate. Okay, and here's example. The sugar is the deoxyribose. You got the base, and um, you got the phosphate. And then so all three components make up one nucleotide. Move on to the next nucleotide, and then the next nucleotide. Okay, and they connected together by phosphate linkage, or known as phosphor diester bond. Okay, how about the bases in DNA? These are bases that contain nitrogens. There are four different bases in DNA. The sugar and the phosphates are all the same. The four different nucleotides only different in four different bases, and that's what we use the letter of the base to call to represent the nucleotide. Okay, all right. Now in RNA, notice that there's no thymine, and that's replaced by uracil. Okay, so urasil's only in RNA, thymines only in DNA. The rest are the same. Okay, for DNA and RNA. Okay, now in order for um, in order to get the correct sequence of amino acid in a protein, we have to have a code, and we only have four bases or four different nucleotides now, but we have 20 different amino acids. So one nucleotide is not going to be enough. So we need more. Two is not good either. So three is perfect. Okay. So we got three nucleotide will code for one amino acid, and we call that a codon. And remember, each nucleotide is only different in a base, so that's what we call bases. Okay, so a codon is a code that has three bases or three nucleotides, and that's for one amino acid. And so that concept was discovered in the 60, early 60s, 1961. Now, for example, in DNA, the AGT code for amino acid serine, and TGA codes for amino acid threonine. So the order of the bases or the order of the nucleotides is very important. Okay, and pretty amazing. The code is the same for whether it's a rat, bacterium, or a human being. It's the same code for the same amino acid. That's pretty nice. Well, it's possible to have more than one code or codon um, for one amino acid. All right. In other words, you can have two different codons that code for the same exact amino acid. All right, here it is. This is on DNA. One strand of the DNA, ATA, that's a codon. Every three bases represent three nucleotides. Okay, and we got the sugar and the phosphate backbone. All right. Okay, what is a gene? Okay, it's a sequence of codons um, in DNA molecules that code for one single protein. Okay, um, and because protein vary in size from 50 amino acids to thousands of amino acids, gene vary too in length from 50 to thousands of codons. And remember, each codon has three nucleotides or three bases. Okay, so you multiply it by three, that will be 150 nucleotides. All right, and each codon, as you see earlier, is a three-letter word that specify one amino acid. And then you connect the codon together, you get to cell gene. And a gene is going to be in one of the chromosome. And in human being, we have 46 of those chromosomes. Okay. So here's the order. All right, we got a chromosome, and we have 46 of those, and come in 23 different pairs. Well, in chromosome, there's DNA molecules, and in DNA, there's many genes, and in one gene, there's many codons, and each codon has three um, different nucleotides labeled by three different bases. And this is the chemical structure of one nucleotide. You got the sugars, the oxyribose, you got the phosphate, and you have the base. Okay, and there's a number for these guys. So this number 1, 2, 3. Remember that's number 3. We call it 3 prime. 4, 5. We call that a 5 prime. So that's why sometimes you see the sequence of nucleotide. You see a 3 prime to a 5 prime or 5 prime to 3 prime. Okay, now DNA is not just one single strand, single polymers of nucleotide. It's actually composed of two for one DNA in a twisted form known as DNA double helix. And the way they twist is really cool. That's the two strand uh, put the bases facing together, and between C and G, uh, there are three hydrogen bonding. And these hydrogen bondings are from the nitrogens and or oxygens with the hydrogens. Now, here it is. This more specific look at the hydrogen bonding between adenine and thymine. There are two hydrogen bonding, and you can see between the hydrogens of the base of one strand bond to the oxygen of the base of the complementary strand. Now, these are not random. Thymine only paired with adenine, or guanine only paired with cytosines, and that's 3 hydrogen bonding. Okay, so they have to line up perfectly. There's no mistake there. All right. So remember, adenine and thymine, 2 hydrogen bonding, facing each other. Guanine and cytosine, 3 hydrogen bonding, facing each other. Okay, what is DNA replications? When cell is ready to divide, and cell division happens a lot in any living organisms to replace um, dead cell, things like that, or to grow. Now, before cell can divide, DNA has to divide first, and that's what we call DNA replications. We're starting out with one DNA molecules. We want to end up with 2 exact copies. Okay, so what happened is when that's about to go, the hydrogen bonding breaks between the 2 strands. So we got two. As they open up, new nucleotide come in and connect together, complementary to the parent strand. Okay, so we end up with 2 copies of DNA. Each contain an original strand of DNA. Original strand. That's new one. That's new one. Okay, so we got 2 brand new, exactly the same. And then cell division happens. Okay, now let's take a look at how DNA direct the synthesis of protein. Okay, in human and animals, um, we must synthesize protein that we need to survive. Okay, and in order to synthesize protein, we need all the components, meaning amino acids. So we have to eat, and we have to eat food that, when after digestions, provide us with amino acids, along with other things. Okay, and then our DNA will reconstruct the, the amino acid in the correct sequence to give us the correct protein. All right. Okay, the first thing is the transcriptions of RNA, messenger RNAs. When there's a need for those protein, the gene will be activated. All right, and when the gene is activated, the that's a segment of DNA. The messenger RNA is synthesized. Okay, so it just open up just like replications, and then nucleotides come in and make the RNA just one side. Okay, now after done, the messenger RNA move out of the cell nucleus into the cell cytoplasm area and work with ribosome. The ribosome is, we'll see the picture, it's a biological structure that contains 2 units and in which the amino acids are connected together to make protein. Okay, so the ribosome move along the mRNA um, and read the codons and then allow the transfer RNA to bring in the amino acid to connect together to make the protein. And here it is. So you got the ribosome with 2 subunits, the large one and the small one, working together, moving along the mRNA, read the code, and then the transfer RNA bring in the amino acid, connected together. And by the time it reaches the end, we got a long chains of amino acid connected together, and that's a protein. And then the protein will come out folded into different structure like primary, secondary, and tertiary and quaternary, so on and so forth. And then the ribosome subunit is released. It's pretty nice how it works. And the codons, uh, the, the first codon will tell um, that's the initiation, and of course the last codon will tell when to stop the synthesis. Okay, so as you can see, um, protein um, synthesis is not random. It's very specific. There are very specific coding for the protein, and that's why each of us look unique because we have different gene or different sequence of uh, nucleotide that code for the protein. All right. Even though we eat the same food, same protein, but we'll look differently because we have different genetic code. All right. So you guys can take a look at the summary here with additional interesting information. All right. Good luck studying, everyone, and let me know if you have any questions. Okay. Thank you all, and have a good day.